All right, thanks everybody for sticking it out to the end. I'm going to talk about some vertebrates at the top of the food chain here, and some of the work that we've done in collaboration with the Illinois Raptor Center. And like John in his research, there are a lot of undergraduates involved in my projects here at Millikan. And so I'd like to acknowledge Emily Mihalkanin, Jess Brinegar, and Shelby Chesko. Each of their projects independently fed into this sort of big picture type presentation that I'm giving here today. So a little bit of background. Fluctuating asymmetry is at the center of the sort of the questions that we're asking with this research project. And fluctuating asymmetry is the development of paired structures that are not in symmetry with each other over time in an inconsistent pattern. And this relative importance really varies across studies, but there was a 2013 meta-analysis that suggested that it is a good biomarker of developmental stress. In particular, most vertebrates, as they're developing early on, they sort of set their pattern for development, and then once they, in this case of birds, once they fledge the nest, they're kind of set with whatever developmental pattern that they had. It's not, they don't really have the opportunity to fix it later. And this is in opposition to the concept of functional asymmetry. There are some organisms that have certainly involved functional asymmetry, like fiddler crabs, for instance, the male having one giant claw to wave in the air to attract females and to battle males, and one tiny claw on the other side. So functional asymmetry is very different from fluctuating asymmetry. And if you're imagining a bird, of course, and you picture the scenario of having, say, one wing that's longer than the other, that certainly can't be beneficial. It's going to add extra work when they're flying, create problems with their biomechanics of flight and landing on perches, specifically with their legs as well. And many bird studies have found that fluctuating asymmetry is specifically associated with nutritional stress in nestlings, but it's also been shown to be a product of habitat disturbance, which suggests that there could also be an impact of environmental stressors on the parents and the quality of the eggs or the quality of the sperm that they produce and the quality of their offspring, independent of the stress that the nestlings themselves face. But comparatively fewer studies have specifically examined the role of anthropogenic toxins on fluctuating asymmetry, and I've presented a couple years in a row here at the Immiquan Symposium, and a couple years ago I talked about lead poisoning. And so because we have the data, I've also pieced together a little bit of what happens perhaps when nestling birds have lead during their development. All right, and some more background. Fluctuating asymmetry is oftentimes considered more within a sexual selection context, but it's also something that can undergo natural selection. And of course, sexual selection, natural selection can sometimes be in opposition to each other. This idea that something that's sexually selected in a male cardinal, for instance, to be very bright red, natural selection may not favor being so bright red because it's easier for a predator to find them. And so this is a feature of asymmetry, and symmetry in particular can undergo both. And even in humans, there's been some suggestion that there's a sexual selection link where people are shown faces of individuals that are thought to be, extremely attractive. And if you skew their face just a little bit and make them asymmetrical, all of a sudden they're rated as less attractive in sort of a quick test, a quick view of their face. And one of the ways that we can link developmental stress perhaps to the developmental patterns that we see in development of anatomy and structures is through stress hormones and signaling along stress pathways. And increased corticosterone, which is the primary avian stress hormone during development in captive birds, does lead to increased fluctuating asymmetry. So carrying out this experiment in captivity is one thing, actually manipulating the stressors in the wild is a whole other thing, but we do know that there are some links between that stress hormone and increasing that stress hormone and developing asymmetrically in baby birds. And further, I work at the interface of stress and disease in most of my research projects, and those of us who work with avian endocrinology are increasingly trying to find other tissues other than blood samples or plasma samples to measure corticosterone. So fecal samples, feather samples, you name it. And the problem with things like feather samples is feathers are grown during a molt cycle in a bird. Unless they lose that feather sometime outside of the normal molt cycle, the only time they're depositing corticosterone into their feathers is when they're actually growing that feather, which is for an adult during molt and for a juvenile or a baby during the nestling period of time. So we're still trying to figure out what feather CORT can actually tell us about a bird's stress profile. So the objectives of the study: one, assess degrees of fluctuating asymmetry in birds of prey. 2, examine relationships between the fluctuating asymmetry and the feather corticosterone levels. 3, examine relationships between asymmetry and parasitism. Thanks to John for the primer for that. And also 4, explore sort of tentatively some links between lead toxicity and the asymmetry that we see in the birds at the center. So the methods, we got to start with the Illinois Raptor Center. This is a wildlife rehabilitation facility here in Decatur. We admit last year, I think the final number was 386 birds over the course of a year. It's a lot of animals and a lot of data. I like to point out to everybody that there's some things that happen to those birds during the time that they become ill or injured that sort of throw off any assessment of their health conditions or their stress, but there's also a lot of things that happen to those animals out in the environment before they became injured. And keep in mind, they were just wild birds yesterday. So when they grew the feather and deposited stress hormones, that didn't happen overnight, and parasites didn't happen overnight and things like that. So there are a lot of good metrics we can collect that aren't an immediate product of their current health condition. Further, raptors are extremely hard to catch in nature, so this is a valuable data source, albeit sometimes a little bit skewed and biased towards birds that maybe are the less fit in general, which I'll talk about later. So at admission, we give them a full physical, we take blood samples, we pull a feather sample if we can, remove a primary or secondary that is either already damaged or harm to them. And this data, the data that I'm presenting today, involves 4 most common species: the barred owl, Cooper's hawk, great horned owl, and the red-tailed hawk, simply because statistical power is present when we have large sample sizes, and these are our most commonly admitted birds. So all of the birds in this data set came from within about a 100-mile radius of Decatur. As you can see, this does include a decent amount of the Illinois River Valley, and of course the Sangamon River cuts right through the heart of this 100-mile radius. So these are birds that are certainly hunting and active within the habitats that we've been discussing all throughout today. When we bring in a bird for an exam, we collect multiple measurements. As you can see here on this slide, I've sort of crudely circled the tarsus length and the wing length because these are the metrics that we use for our asymmetry scores. We take 3 measurements and then make the recording, and then we also then turn around and calculate an asymmetry score, which is the absolute difference between the wing lengths plus the absolute difference between the tarsus lengths. And we divide that by the average wing length with the notion that a bird that is very large, a couple millimeter difference is not as big a deal for a bird that is very small. So that's some way to correct for the fact that larger amounts or small amounts of asymmetry might be more meaningful in birds of different sizes and vice versa. So the 3 parasites that we surveyed for using blood smears are Plasmodium species. If you look at this picture, you can see the Plasmodium parasite, as it develops, actually pushes the nucleus to the side. So my cursor right now, if you can see it, was actually on the little developing parasite. Hemoproteus species is also another form of avian malaria, but it actually sort of wraps itself around the nucleus and doesn't displace it. It's sort of represented by the baby blue colored parasites inside these red blood cells throughout the slide. And leukocytozoans infect white blood cells, primarily monocytes, and they produce this elongated shape that looks like sort of a 2-tailed shape structure over there on that screen. And they're the easiest to identify, certainly, but they're a white blood cell parasite. Any of these are generally classified as avian malarias in multiple papers and studies, and these can be routinely surveyed through blood smears when they reach that mature phase. For feather corticosterone measures, again, this is something that's usually measured in blood plasma. It is a stress hormone, but it is deposited in the feathers during growth and molt. So from a method standpoint, we extracted the corticosterone from feathers using a method that was developed by Christine Latin in 2011 with red-tailed hawks specifically, where we extracted the corticosterone from feathers using methanol and sonication. And then we used an Arbor Assay's corticosterone enzyme immunoassay to quantify the amount of CORT in samples after reconstituting them with the assay buffer. So getting into some data now, we found a significant interaction between the relationship between asymmetry and feather corticosterone between age and asymmetry, because basically we had no relationship with adults and a strong relationship with juveniles. And so you can see the R squared for the adults is 0.016, it's almost a flat line. Whereas we see about 0.326 strong positive relationship here between asymmetry score and feather CORT in juvenile birds. And you'll notice that there are almost no adults up in the upper right quadrant of the graph. And we'll talk about that in a minute. We basically think that this is evidence that we're getting a lot of the lower quality juveniles that are ending up sick and injured in the Raptor Center that perhaps were in bad shape from the start, and adults just don't survive to be high stress, high asymmetry birds, as they don't make it there. From parasitism standpoint, we found that for every 1.48 unit increase in asymmetry, birds were about 4.5 times more likely to have Hemoproteus infection, independent of species, so they're all lumped together here as they were in the previous slide. And slightly less strong, but still there, for every 0.744 unit increase in asymmetry, birds were 2.17 times more likely to have leukocytozoans. So this suggests that after being stressed early in life, perhaps as they progress through life, the early life stressors might also have led to immunosuppression, and the persistent stress associated with asymmetry might also be immunosuppressive, leading to an increased risk of parasitism, less likely to be able to defend from parasites with their immune defenses. Also, if we look at the symmetry difference between the age groups, you can see that the juveniles that we admitted to the Raptor Center during this time, about 48% of those birds were on the asymmetry scale, and in adults, it just doesn't happen that often. So again, we see a survival difference, and it does suggest that some of the birds that we are rehabilitating were already in very poor condition, perhaps from the start. Looking at lead poisoning, this is something that we have monitored from all of our birds for the past 4 years. This is a fairly well-known problem for eagles and vultures, but we don't really know about those species that are rarely scavenging. The literature is a lot more sparse on those. From the eagles and vultures that scavenge, it's well documented, especially in the upper Midwest, that gut piles that still contain pieces of lead shot, upland game, deer gut piles, so on and so forth, are a common source of lead poisoning. But we've actually found Cooper socks and eastern screech owls, which are commonly found in urban areas and also eat other birds and other small prey, to commonly have relatively high lead levels as they come in. And this is something that we started testing urban prey, and we actually found that small avian prey species like rock doves and house sparrows and robins and European starlings are also typically high on the lead detection scale. So it's possible that they're just getting it from the urban environment in which they reside. Regardless, if they're exposed to lead at a very high level frequently, then the question is, does this impact their offspring, and does this play a role in the big picture I've talked about here today, early life stress and fluctuating asymmetry? And so if you look at this picture, this image, we did find an age by lead significant interaction. Again, the adults, there's no relationship between asymmetry score and whether or not they were lead poisoned, but nestlings do get lead poisoning, probably because they're fed poisoned prey from their parents. And you can see that there's much more asymmetry associated with the individuals that had detectable lead levels well above baseline. So if you look at the bottom, the x-axis here, we have under 3.3 micrograms per deciliter, which is the undetectable level through our machine, and above 3.3, which is the detectable level in our machine. And we have a significant difference in that juvenile birds that have detectable lead are also consistently asymmetrical, at least compared to those that don't have lead and compared to adults. So take-home message, juvenile raptor asymmetry shows a positive linear relationship with the stress hormones deposited in the feathers while they're developing, and that was uniform across the 4 species. This was also not significant, and there was no relationship there in adults. So this is actually a pretty good validation of the link between early life stressors when they're growing those feathers and depositing those hormones, and that it does, in fact, influence symmetrical development. Asymmetrical raptors were far more likely to harbor blood parasites than the symmetrical birds, and this was independent of species and true of adults and juveniles, but stronger in juveniles. While lead only has affected about 1/5 of the birds in our study to 1/4, blood lead levels were correlated with fluctuating asymmetry of juveniles, and keep in mind that about 48% of our juveniles were asymmetrical. So again, toxins in the early life development might also be impacting the ability of these birds to develop in a symmetrical way and have long-term influence on their likelihood of survival and achieving fitness. And finally, we do have limited data from raptors that are not brought into rehabilitation centers to know if these percentages are consistent across individuals, but we are currently amending my banding permit and going to start doing more trapping of sort of non-Raptor Center raptors, so non-rehabilitation raptors, to get a better sense of how well our data match those that are just free living out there in the wild that haven't suffered from some kind of injury or illness. So with that, I'd like to thank the Champaign County Audubon Society, Decatur Audubon Society, and Millikan University for funding this research. Most of that went into funding the lead poisoning work. The Illinois Raptor Center, the volunteers who take care of the animals on a daily basis and feed them and make sure their cages are clean so that the rest of us doing the scientific work have healthy animals, or improving health animals to work with. The donors and also the board members who approve of the work. And then my host institution, Millikan University, for continued support for this research and the research of my undergraduates. So with that, I'll take questions.
Thank you, Travis. I do see a question has come in, and I'll turn things over to Doug to moderate that.
Thanks, Tom. Thanks, Travis. First question is, does the researcher have any ideas about the source of the lead in those birds, the non-scavenger birds? I guess it is, yeah.
Yeah, so we've spent a lot of time trapping, in particular, small songbirds that live in the areas that we're getting these raptors from, and consistently finding that the sparrows and robins, starlings, and pigeons in particular come in with a more frequent detectable level above baseline than even do our raptors. So we think that they're getting it from urban sources. Those urban sources could be, non-remediated paint issues, old dilapidated buildings. Of course, we're a major industrial city here in Decatur, so I know there's a lot of lead waste that's probably a byproduct of many years of using it for many different things. And so we do see those links in the non-scavengers are much tighter in the urban birds than in the birds that live more out in open areas. So I suspect songbirds and maybe small mammals, if we were to trap them, and specifically just a product of human waste products.
So I was wondering, do you have an inclination about what you'll find in those free living birds?
I do think that we'll find the same relationship, but I think our average, when we go out and capture free living juvenile birds, for instance, I think we're going to find a skewed percentage of symmetry back towards more symmetrical birds.
Yeah.
I do think that we're seeing what's happening is we're rehabilitating a lot of birds that may have a tough time in nature anyway, because they are sort of the lower quality individuals. That doesn't mean we shouldn't rehab them, because obviously there's conservation issues with top predators, and maybe they make it after the help we give them. But it does certainly, that data certainly suggests that we're getting a lot of pretty low quality birds in general, juveniles that were just not making it well in nature in general.
So I have a quick question, Travis. When you're saying that they're asymmetrical, how much is it? So if we're comparing the left and the right wing.
Sure. So extreme cases, I would say sort of if you look at the range, we had some birds that had, we had one red-tailed hawk juvenile that had a 12 millimeter difference in wing length and an 8 millimeter difference in tarsus length. So that was sort of on the high end of the spectrum. And again, that's not a lot if we just put it to a ruler, but I think given the biomechanics of flight and landing, that's probably a big deal, even for a large bird like that.
Right.
They fly in circles all the time as a result of that, I wonder?
That's what I just keep thinking, right? Like if you build an airplane that way, it's just going to spin.
Here's another question. Does the gender of the adult or juvenile have an impact on the degree of asymmetry or the level?
Yeah, so it did not. In the data that we presented here, I didn't present those data in particular, but we did do that analysis, and it has not influenced. The other problem, of course, is some of the juveniles, we don't know their sex yet. So in the adults, certainly no. In the juveniles, we only know that for some species. So it could, we can't definitively say with the juveniles.